Okay, hello. So I'm Mia Krout, and I'm going to talk to you a little bit about how we have applied high pressure freeze for TEM and looking at doing single cell reconstruction in immunohistochemistry, obviously using C. elegans. So first, I'm going to tell you why we use high pressure freeze EM. We all use high pressure freeze EM, so I'm going to give you a little bit of background about it. Here's just an example of a classical chemical physical fixation versus a high pressure freeze physical fixation. Both of them are looking at the ventral nerve cord of the worm. And you can see, is there, okay, yeah. So you can appreciate the fact that just here the neuronal membranes are a little bit ruffled, maybe some shrinkage occurring here in the neurons, whereas the round sort of plump neuron seen here using high pressure freeze fixation. These are the aesthetic differences that you might notice based on just the actual differences in how these methods work. The classical chemical physical fixation takes many hours versus milliseconds in the high pressure freeze method. And this kind of, the changes over time in the molarity result in sort of these changes that we might see in the ultrastructure. So you might end up with an impacted ultrastructure preservation versus what we think is a more near native physiological state using high pressure freeze. That's not to say that the classical chemical fixation method isn't useful. It very much is. This just depends on the question you're asking and your area of interest. I'm going to show you an example of classical fixation, an early study done looking at UNC13 null mutants, where the conclusion was reached that UNC13 was acted downstream of docking, and this was based on the fact that UNC13 retained docked vesicles, where the same group used high pressure freeze to answer the same question and found a very different result years later, showing that UNC13 null mutants actually lose a large part of their docked vesicles. The findings of this study have actually been recapitulated many times and have been shown to be true in other systems. So now I'm going to give you just a little bit of overview of the way we actually use high pressure freeze in our lab. I'm kind of giving you a basic introduction to the technique. In general, we are all going to be using different equipment, I'm sure, and different methods to get our samples prepped, but this is kind of what it looks like for our lab. So you put your worms in a specimen carrier and you use a cryoprotectant. That's OP50 with a tiny bit of water. We use a Leica HPM freezer, and then this freezer freezes the worms instantaneously at really, really, really low temperature under really, really, really high pressure. And so we remove those worms and put them into liquid nitrogen. We put them into cryovials until they are actually unloaded on our really handy unloading station that our machine shop made. It's sitting in a styrofoam cooler, and we fill it with liquid nitrogen so that we are maintaining really low temperatures during this unloading. And so we remove the samples from their cryovials and we put them into their own individual inserts in these spider rings, and those universal chambers get put into our Leica AFS. That is an environment that's really temperature controlled and allows for us to do our, to complete our fixation and do our substitution. And here is the interior of this cool chamber and the program we use for our AFS. We use tannic acid and osmium tetroxide for our fixations and substitutions. And then we embed our worms in EPON. And so we basically go from this substitution of tannic acid to osmium tetroxide, and then we do a slow slow infiltration at room temperature of the resin acetone mixture up to more resin, less acetone, so on and so forth, until we remove the samples from their carriers and we put them in EPON. And this is my least favorite part. Here, we actually pick the individual worms from their bacteria. It's terrible, and it involves really, really tiny needles. But we care about the orientation of the worm once we're sectioning, so this is a step that is important for us that hopefully you don't have to do yourself. And then we actually place those individual worms onto a drop of EPON on a glass slide, and we put a few pieces of parafilm between the bottom glass slide and a top slide that's heavily coated in Teflon. And these cook for at 60 degrees in an incubator oven for like 2 days, and then we remove individual worms from this thin embedding and mount them on these resin blocks also made of EPON. And we then use, we do some rough trimming, but then we use diatom knives to achieve this trapezoid-like shape that is helpful in creating a ribbon you see here that we collect on carbon slot form bar slot grids, and then we image them, and that's the nice part, right? Because then we get some pretty pictures. And here you can see we have used this technique to primarily investigate the nervous system. Here looking at either the dorsal cord or ventral cord, depending on your question, and we look at things like docking, docked vesicles, dense projection, the distance of docked vesicles for dense projection. But basically, we acquire a lot of these sections of the synapse, and from there we use ImageJ, Fiji. There's a plugin, TrackEM2. And you can use TrackEM to align your individual sections. Here you can see I'm aligning 2 sections so that I can keep my region of interest in the same plane moving forward. And you can actually just use TrackEM to mark up all your sections and create volumetric data there. Like this example TrackEM has of its usability of these neuronal arbors. But you can also use, well, many softwares, and I'm certain you'll hear about plenty of programs later in the talks, but you can also use a program 3D mod, which is frequently used to process tomograms, like the one you see here. This one is about 200 nanometers thick, and you acquired this using TEM, but a tilt series. And this way you can you can get volumetric information from a single section, but because your sections are thicker and you're tilting, you're able to sort of create this, look at a single synaptic profile, but get more data on the volume there. But you can also use your ultra-thin sections. I primarily section 40 nanometers thick, and you can actually use that to your aligned sections to get your 3D reconstructions or volumetric information at a single synapse, which is what we have historically done. But recently, we were working in collaboration with David Miller, and we were more interested in instead of a synapse, we wanted to look at changes of a neuron, a whole neuron, a single neuron. So you can appreciate here that what I've done is exactly what I showed you, just a lot more sections and a terrible, terrible amount of time, and put these together and aligned them and marked up the areas of interest to get this single neuron and be able to look at these in a wild type and a mutant animal, and look at, we just were able to get information on a single cell level versus looking at these individual synapses. moving forward into something we're working on now, we're actually looking at a different single cell, a selenocyte. And thanks to Warm Atlas for this picture here. But the selenocyte is used as a cellular model really frequently to look at the endolysosomal pathway, and that is what we are also using it for. And so we were able to create this model of a selenocyte and its endo and lysosomal compartments, as well as the transport vesicles surrounding those compartments. And we were, in this study, we're also using fluorescent tags, right? So we're tagging all of these proteins involved in the endolysosomal pathway with GFP, and we're looking at these expressions and these organelles in the selenocyte, and we're putting together our EM data, and we're marking up these organelles based on essentially just their physical characteristics. And then we felt that we couldn't really be confident about how we were, maybe we weren't, maybe we're missing something, right? So we turned to a second EM technique. And here is an example of looking at subcellular protein localization using immunogold beads. So this is a very useful technique. And it starts in the high pressure freeze, very similarly to what I showed you previously. But the chemical and substitution vary just a little bit. Instead of tannic acid and osmium, we used uranyl acetate. And then we actually embed these in HM20, which when it polymerizes, it's clear. And the worms are fairly clear. And we don't remove those from their bacteria. We just mount them and section them similar to our ultrastructural studies, but we don't have to do the picking out of the bacteria that time. So yeah, here is a proof of concept. Since we were going to be looking at GFP tagged strains, I looked at PMyO2 GFP in the pharynx, and here you can see hopefully that we were able to, we have really great antibody specificity here in the pharyngeal muscle cells with no signal in these adjacent neurons or in the surrounding tissue. So we're really pleased with that protocol. And we use that to then look at how our various GFP labels were, we're marking our proteins of interest. And these little red dots are actually trying to help you see the immunogold beads. But this is just an example of how we were able to use our immunoEM to tie together the fluorescent data with our volumetric reconstruction. So you're going to hear about a lot more elegant ways to achieve this volumetric data, but just using my same techniques and my same equipment, I was able to get some really beautiful and meaningful data out of it. But also just a reminder that there are a lot of EM techniques and you can combine them to do some really cool stuff. So yeah, thank you. I'm at University of Illinois Chicago. I also published a protocol on the basic ultrastructural data. You can look that up and you can email me if you have any questions. Thank you.